Our topic is B.8 and it's about nucleic acids. And what we know about nucleic acids are that they are DNA and RNA.
Woohoo! So, first objective, there are only 5 main objectives in this topic. First objective is P.8.1, which says describe the structure of nucleotides and their condensation polymers, nucleic acids, and polynucleotides. So the first thing we need to know is what a nucleic acid looks like. There are 3 main parts to a nucleic acid. First, there's the phosphate group. Then, there's the sugar group. And then we have our nitrogenous base, which is, we're going to label as B. So, we have to split it up into the 3 parts. We'll explain each one individually. We start off with our phosphate group. Phosphate group has a very simple structure with a central phosphorus, double bonded O, and then 3 single bonded O's with electrons. That's the phosphorus group. That's the beginning of our nucleic acid. That is then bonded to a sugar group, which is different for DNA and RNA, but that will be touched upon later in the presentation. And then finally, there's the nitrogenous base groups. Nitrogenous bases are split up into 2 parts, purines and pyrimidines. The purines are adenine and guanine. They are 2 rings, while the pyrimidines are cytosine and thymine, which are single ring. You see the uracil down here. That is unique to RNA. So DNA would have adenine, guanine, cytosine, and thymine, and then RNA would have adenine, guanine, cytosine, and uracil. So we said earlier that a nucleotide is made up of a phosphate group, a sugar group, it's not really sugar, but, and a nitrogenous base. Now how do these bond together? If you look in the objective, it says the condensation polymers. So there's condensation here as well as in other places. So the first place we see condensation is between the phosphate and the sugar. Right here, to make this linkage, there will be an H2O released right here. And the same thing between the base and the sugar. Dehydration, synthesis, or condensation releases another H2O. Now, how do these nucleotides bond together, you might ask? Because a DNA isn't just one nucleotide. It's many nucleotides packed on top of each other. This happens through condensation again. You have your sugar, your phosphate, and your base. They are bonded together. And then the phosphate group, through condensation, again releases another H2O to make this bond. And then this goes so on and so forth. Alright, our second objective is B.8.2, which is distinguish between the structures of DNA and RNA. The first thing we have to account for is the sugar. The phosphate group is the same in both, but the sugar is different. In DNA, deoxyribonucleic acid, we have deoxyribose, which is this structure with an H down here. Now if we were to change it to ribose, which is the sugar in ribonucleic acid, or RNA, we take off that deoxy, we take off that H, and we replace it with an OH. That is the sugar difference between DNA and RNA. The second main difference between the structures of DNA and RNA we touched upon earlier when we were talking about the nitrogenous bases. I don't know if you remember, but there was that thymine uracil difference in the pyrimidines, and that's about it. Now, the strand as a whole has a main difference. There's something called the double helix, and a single helix, I guess. The DNA is a double helical structure, while the RNA is just single-stranded, so it's a single helix structure. Third objective is written up here. Earlier we talked about the double helical structure of DNA, and that's what we're talking about now. DNA strand, as we said earlier, is two strands put together. First strand is the red strand, second strand is the blue strand, and each strand has its nucleotides on it, which are represented by these little hashes. We said earlier about the nitrogenous bases, and over here each nitrogenous base pairs with its with its corresponding base. First pair we have for DNA is the adenine and the thymine, which is AT, and the second pair we have is the guanine and the cytosine, which is CG. And these bonds, or this interaction, is what we know as a hydrogen bond.
Our next objectives involve in protein synthesis, and it is the creation of a protein, and it starts from the DNA, and transcription. It is transcribed to create the RNA, and that has a special code, and through translation the protein is made. And here we have our DNA strand, and in translation the strand is unwound, and after that there are specific codes that is created by the sequencing of the nitrogenous bases on each strand, and that allows for specific enzymes to attach the complementary base to each of the specific bases and have a code. So essentially the DNA is the template to the creation of the mRNA in transcription. So I'm going to now describe the process of translation. And so our mRNA that was from the transcription process has left the nucleus, and it goes to a ribosome, which is also an rRNA, and each three bases are tripled together, and they're a specific codon. And when it's in the RNA, the tRNA brings specific anticodons that are complementary to each codon, and each tRNA has a specific amino acid that it brings. So as the process of translation keeps going, each three is brought with more and more amino acids, which are bonded together by peptide bonds, and as the process ends, there's many peptide bonds and many amino acids, and that creates our polypeptide and thus our protein. And that brings back to the whole process, the central dogma of how the information comes from the DNA to eventually end at the protein. And so the DNA provides the information to create the RNA, which then provides the information for each amino acid that goes to our protein. So if we ever have a question of the identity of an individual, we can use DNA profiling, and that is extracting the genome of the individual. And so first we get a blood sample, this is blood, from the individual, and the DNA is extracted from the blood. And this DNA is cut by restriction enzymes into short tandem repeats. And that is the first step. Now, moving on, we have the second step, and it is the polymerase chain reaction, and it allows for these short tandem repeats to be multiplied at a very fast rate. And that allows us to create millions of copies in a short amount of time of these repeated strands. And we can then load it into jelly electrophoresis, which is the third step. And in jelly electrophoresis, each of these strands are loaded into the negative end, which allows for each of the strands to travel towards the positive end. And you may ask why? And it's because the nucleotides within the DNA strands have phosphate groups, and each of the phosphate groups is the reason why it contributes to the negative charge, and thus the negative is attracted to positive, so it goes towards a positive charge, and distances vary through the size of the strand. And the uses of DNA profiling are victim identification, forensics casing, and biological relationships, and to determine relationships between population, which include evolution, ecology, and migration.